Hello and welcome to SciLux, the podcast where we talk about scientific developments and technological changes in Luxembourg. Most of the times, right now in this season, the new season 5, we'll be also talking to scientists from outside of Luxembourg. But today is the first episode, proudly powered by Research Luxembourg, recorded at the Media Centre of the University of Luxembourg, which means we're talking to a scientist based here in Luxembourg. And the scientist is Professor Anne Grünewald. Who is Anne Grünewald? Well, she's Professor of Neuroscience and the head of the Molecular and Functional Neurobiology Group at the LCSB, the University of Luxembourg. Her work focuses on mitochondrial biology. Anna, thank you so much for coming today.
Hi, thanks for inviting me.
I think whenever I talk about biology or related subjects, I admit in the beginning that I hated biology at school, so I have to say it again. This was really one of the subjects that I really didn't like. There is something about schools, education, biology and all that. And mitochondria, I remember that it was something, I don't want to do this. How do you end up getting passionate about mitochondria, actually?
It's actually funny because it's for the exact contrary experience. So I had not really in the last 2 years, but up to, I think, the 10th grade, I had a really extraordinary biology teacher who really made me passionate about the subject. And I guess even at that time, I knew that I would like to do something related to biology. So indeed. So I guess it all comes down to the teacher. How was your teacher?
Well, I will not mention any names or anything. No, it's just that, listeners know I'm originally from Poland, and somehow biology was always just this learning by heart that I just couldn't do because maths was perfect, something else was logical for me, but this was just really lists and lists of things and functions, and they just didn't stick.
She somehow created some kind of environment which made this really dynamic. It's true that obviously it also had a lot to do with just learning, but I guess she connected the individual processes in a way that it also had some logic behind. And I suppose I also liked it in a sense that it was a representation of life in general and trying to understand how life works in a way.
And this is a great point because I think, when you look at the podcasting scene right now, definitely biology, life, medicine, and neuroscience that we'll be discussing is a fashionable subject, probably because it relates more to us. And it's easier to find audience that can relate to it, I think. I don't know if you would agree with that.
Exactly, because I guess if you learn something there that you can really apply to your life and maybe that improves your health, let's assume, then, you can relate to these topics, I guess, easier than, let's say, if you were to discuss some complicated topic in physics. I mean, just assuming that if you can then transfer this information and somehow implement it in your own life, and if it's even something as simplistic as realizing that your lifestyle and sport activity might have an impact on whether you will end up with dementia or not, that I guess has a higher implication in everyone's personal life than, I don't know, knowing how bridge XYZ has been constructed. But
I guess some engineers would disagree here.
Yeah, I'm not saying that that this is irrelevant. It's obviously highly relevant, maybe in fact more relevant, but in a sense that for the personal use of a regular citizen, I guess they can make more use of this information.
Yes, true. But, I was also thinking, looking from my perspective of science communication, is that somehow there are a lot of people who manage to make it a bit clearer than other fields of science. And I was wondering, it's not that in neuroscience you can really say A is B. It's science, so it also depends. So when you listen to people, I have to mention it, of course, Andrew Huberman. When you listen to him, he is a little bit A is B. How does it make you feel as a scientist then?
One has to point out that his facts that he hands out to the public are really based on scientific literature. So that from that aspect, I find it great. So it's not like he just shares opinions in a way. And he also always provides the scientific background. My problem is, however, that these podcasts, it's still selective. It's still selective about what kind of literature you want to highlight. So in that sense, it's great, I guess, as an entry point to understand a certain subject that sometimes maybe even I would not understand otherwise. But I suppose you should then just take it as an invitation to dig further yourself. And I'm not entirely sure that this is what people do. I guess it's often seen as the answer to whatever question. And that's maybe a little bit a concern I have with podcasts in general, if they are kind of trying to digest a certain topic for the public. But then again, that's a criticism. But on the other hand, I actually feel that without such media, it wouldn't be possible to make people even enter these fields. So I guess it's also still necessary and to somehow provide them with some information that is exciting, that is maybe even catchy, and that they can indeed somehow apply to their life, because otherwise it just stays like a box that they may not even dare to touch or to look into. So yeah, so it has its pros and cons.
In general, also when I prepare for the interviews, it's always 2 weeks. It's a completely different field of science, right? So I try to get as much context as possible, but of course I'm unable, and I'm really reading the scientific literature and preparing myself. So I don't assume the listeners will then go and read all the stuff that I read, although I share most of it in the show notes. Still, I guess it's better than, saying, there's no point. I mean, we're still talking, we're giving context. And, the context I'm lacking as well. If I ask you now about your research, I just read those few articles, but I don't know in which context you're doing your research and what it means for the fuller picture.
Yeah, and the question is whether this is what I would expect from a podcast to be able to do that. I guess not. I mean, there has to be a limit also simply time-wise. These episodes are already relatively long. Let's say if you're a working mother or dad and even have a family to listen to this at the same time, it's not that simple. So in that sense, I completely understand that there has to be a sort of selection going on. Just that we are all biased in our selection, and simply this awareness maybe is critical. So that not even maybe so much that I would like to change the content, but to make people very much aware that there always has to be a selection, and that this is obviously driven by whatever idea certain scientists supports. That's also what I have to do in my everyday life. I have to pick the topics I want to understand better, and already there is a bias, because I may like to work with topics relating to mitochondria, so I'm biased to, and I know that they have something to do with neurodegeneration, so I'm obviously biased towards what the role of the mitochondria is, whereas there's also the lysosome and there is the proteasome, and all these systems are equally possibly involved, but my heart beats for mitochondria, so obviously I'm gonna look in some ways, not totally selectively, but I'm going to spend more time investigating this. And that's also a little bit the same picture I see in these podcasts. I mean, obviously, Uberman himself has done research. He might be then also influenced by his own previous work, how he picks the topics and who he knows, who he meets, and who he gets somehow excited about.
So let's look at your topic selection, right? So first of all, of course, I researched your biography and everything, and I noticed that you started with biotechnology and then switched into neuroscience. So my question is, how close are they? Are they related?
Maybe first, how did I get into biotechnology? That was kind of maybe as mostly pragmatic choice, because biology itself, while I really loved the subject, but I didn't at the time also really understand what can I eventually really do with that, because it's rather broad. You may end up in some specialization depending on where you are in something like zoology or what really I didn't care about. And at the time, I guess I was mostly excited about anything genetic. And so biotechnology sort of promised to be exactly that, to be biology with a focus on, let's say, new technologies that can be applied to genetically manipulate. Not even so much that I had the plan that I wanted to do this, but I wanted to understand the technology behind that. And on top of everything, I just have to say it was also simply in fashion at the time. So I graduated from school in 2000, and biotechnology was really that thing. But already when I started studying this, I realized that I totally underestimated the engineering component in this. Not that I wasn't able to do it, but it was simply not so much my main interest. So I actually graduated with an engineering degree. You can do that in Germany. It depends on the university where you graduate, but mine was a University of Applied Sciences. And the university itself had previously only offered like real engineering topics, and biotechnology was kind of the new cool thing to put on top. And this is also how this course was made. But as I don't know, from how my family worked and so on, like I kind of learned to not give up just because, so, okay, if I really couldn't have passed certain exams, that's the reason. But since I still agreed with the overall aim of getting this degree, and even though I may have not liked probably something in the area of 50% of the lectures, but as I was able to do it, I just went with it. But then again, I don't want to sound sad, but like when you come from Eastern Germany and you've somehow seen your parents struggle and everything was complicated, and now you are finally out there and you were somehow responsible for yourself, and you're supposed to finish these studies in a sort of efficient manner, then you don't now start debating over whether it's sensible or not to have topics like whatever Signal- und Systemanalyse or some stuff like that. Then you just do that. And that was a little bit the approach. Yeah, and along the way, I already knew that I would, if possible, do a PhD, and that maybe this would give me finally the opportunity to really pick something I really, really, really wanted to do. I was in a way lucky because coming from this engineering background is actually not so straightforward in Germany to do a PhD. So I had only few universities that would even offer that to people from this type of university, which was a University of Applied Sciences. And I was simply in a way lucky that I only really applied in 2-3 places, and that it turned out to be the University of Lübeck and the Institute of Neurogenetics, or at the time not even institute yet, but a section for neurogenetics, something like that, that they were somehow open-minded enough that even a biotechnologist can do something there. And this is how I kind of got onto my path.
And you said, okay, now I know it's neuroscience, or not really. Because, I keep pushing for this, but it's also interesting, I think, for people who are choosing their paths, is that it's absolutely not linear for most of us.
Oh, not at all. Not at all. Nothing was linear about it. I guess it was a little bit like this, still this fascination for genetics, which was maybe not sufficiently served by these studies, I have to say. And maybe also a little bit the motivation to move away from this really core biotechnology topic where, especially at this university, it was very much like, you want to then produce something in industry and so on, and maybe pharma, biotechnology, a little bit into that direction. And maybe it was almost a little bit that I specifically don't want to do that. And the brain has always been fascinating to me. So then to move into neuroscience seems maybe obvious. And as during that period, genetics in that field, and here in this particular lab, it was all about movement disorders. That was really moving. That was a phase where with genetics, loads of new things came out all the time, because that was still kind of early-ish days. And simply by just doing genetic analysis in a family, you could quickly find the cause of the disease. I also thought that this was super exciting. I mean, now we know a little bit better that just finding a gene doesn't really mean that you've answered all the questions. But that was still a little bit a phase where everyone thought so. And so this had a huge potential for growth. And indeed, now this same lab is its own institute. So that did work. And I guess I liked the spirit.
And it's true that I guess at that time, genetics was great for the dreamers because it was really developing so much. Let's move to mitochondria. First of all, what is it really?
So it's commonly declared the powerhouse of the cell, the mitochondria. So that basically means a little bit like in a powerhouse that all of these nutrients that we take in, they have to be at some point converted in an accessible form that can be used by the cells to basically serve their energy needs. And this is exactly what mitochondria do. So they convert, in a whole chain of processes that we call metabolism. They end up producing ATP, which is an energy equivalent that can then serve almost endless amount of processes that are needed for different cells to do different functions and so on. So this is what the mitochondria do. So typically, when you think back to your school knowledge, they are these little bean-shaped organelles, which is already wrong because they are in reality not little beans, but they're more like a tubular system that constantly fuses and undergoes fission. And so they are quite dynamic, and they're really kind of spread throughout at the cell, really more like an information network almost. There's actually also a term which is now commonly used in this field that is mitochondrial signaling, because they are not just these producers of this energy equivalent ATP, but they do so many more things as a network. And that's what I find so exciting about them.
Listeners, if you want to see something as well from what Anne is describing, I invite you to go to YouTube. You can also see some videos where she's presenting her work that was a couple of years ago with the Attract Fellowship of FNR, but still you can check that out. And that, of course, the videos, when I see something, it always makes me think about the color as well. Because I think, in scientific illustration, we have to choose colors, but they are not always really real, right? So what's the color of a mitochondria, actually?
So in my heart, the mitochondria are green. But this has to do with a commonly used dye, a mitotracker. It's called mitotracker green, which allows simply staining mitochondria in a green fluorescent way. And actually, it's a cool topic, because I had exactly that topic with some of my mito postdoc friends in Newcastle at the time. And we all had like a specific antibody that we preferred that would label the mitochondria. And each of us had a different preferred color of the mitochondria. So goes to say that they don't really have a color. So it's really mostly in our minds that they have a color. But like for me, if you ask me, it would need to be green.
Okay, so you can really choose the color in the end. I mean, there's not only one type of...
So it may either depend on the dye, or if an alternative way of labeling them would be to pick a protein that is quite abundant in the mitochondria. So we often use a protein that's situated in the outer mitochondrial membrane. And then basically the system works like that you have a first antibody that binds that target protein, and then another antibody that binds this first antibody. And so this secondary antibody, you can more or less define the color with this secondary antibody. So that means within a certain range, your color choice is up to you. But yes, for me, it would be green.
So if you see them diet in a different way, start protesting. I'm not reading this article.
Well,
not that much, I assume. It's still about what is inside rather than the illustration, I guess. What about talking of illustrations? When I went to your LinkedIn profile, you also have a picture there. What is the picture of?
So these are, if I remember correctly, these are dopaminergic neurons, or let's say a neuronal culture with dopaminergic neurons as one of the subpopulations. And that's obviously because we are working on Parkinson's disease, where those dopaminergic neurons are the neuronal subtype that is the most affected or vulnerable, if you like, and where we're trying to understand in my lab what could be the possible influence of mitochondria in this vulnerability of these dopaminergic neurons.
We'll move to that in a second for sure. But, just listeners, be aware that if ever Anne changes into some illustration with something green, that's probably some mitochondria.
But I actually don't think that in this particular picture that there are mitochondria inside.
No, for sure there's no green. I mean, I don't know what you would choose, but...
This was actually taken by one of the first PhD student that I had here in Luxembourg, so Kobi Wasner, who is from New York and back in New York now. So thanks, Kobi, for the beautiful picture.
We said a little bit about mitochondria. We're going to continue because I want to know obviously more. And one of the things I want to know is obviously the genome and everything related to that. But I know that you prepared a pub quiz question about it. So let's first ask the question and then you tell me everything else except for the answer to the question, because that's only at the end of the podcast.
So my question is for the listeners, if they have a rough estimate of how many copies of this little circular genome that exists in these mitochondria they assume would be present per cell.
And we're talking about estimates, of course, because
Yes.
as usual with this, we can't just give an exact number. So listeners, please remember, answer only at the end. So wait a second, genes in mitochondria. So we have a cell. And how many genes do we have there? And can we just only look at the mitochondrial genes, I guess? And they are important for what? What do we know about them?
So what's kind of super special about the mitochondria, at least when we consider the human body, is that this is the only other location where you find genomic information outside of the cell nucleus. So all other DNA is contained within the nucleus. And here in this particular case, these bean type of organelles, they do have yet again their own DNA, and it's circular. And it's reminiscent of DNA that you would also find in a bacterium. And that is, these days, we explain that through a certain theory, which goes along the lines of mitochondria having been, let's say, bacteria in the past and having been engulfed by cells in a symbiotic manner. So basically allowing the cell to function more efficiently and also the mitochondrium to function more efficiently, and thereby basically the mitochondria, including this circular piece of DNA, remained part of this host. And it's actually curious because we cannot, so first of all, there is this little genome, but it doesn't encode sufficient numbers of proteins for the mitochondria to function. So the mitochondria are highly depending on proteins that are still these days now being produced by genes that are encoded in the nucleus, and then these proteins have to be imported. And it's also goes the other way. So in the nuclear genome, you can find that most likely had an origin in the mitochondria. So the DNA has basically moved into the nucleus. So that's really highlights how interconnected this system is. And so for the mitochondria to function, there has to be an import of about 2,000 proteins all the time from the outside so that they can basically do their job. And only, if I'm not mistaken, 13 proteins are being produced by this circular DNA. There is also lots of machinery encoded by this little circular DNA molecule that is needed to actually do transcription and translation of, yet again, this little circle. But only 13 of these genes that are there are producing proteins.
And what is the typical size of mitochondria? Difficult question probably to answer.
Of the mitochondrial genome or
of the mitochondria
themselves? Oh, God. No idea, because the problem is there isn't a fixed size. So it's a network that spreads throughout the entire cell. So then basically the cell size defines, in a way, the mitochondrial length, if you like.
Okay, I get it. So it basically means it's not like you have it in one place, but it's spread around.
It's spread around. So you can see this very nicely in cells that are quite spread out. So for instance, back in the days, we did a lot of work in skin cell-derived cell cultures, in fibroblasts, and they are quite flat and very spread out cells. And there you would find the mitochondria basically going along the entire shape of these cells. And then how do you define then a mitochondrium? Is it a whole thing, or is it, and also if you assume that the mitochondria are constantly fusing and then dividing again and so on and so forth, so then you would need to say at this very time point, you could potentially calculate the size of the mitochondria and then make an average. But I find it's a bit arbitrary because really that goes a little bit against the real function, which is kind of this tubular network that exchanges all the time content and proteins and also DNA itself.
What about dysfunction? What happens if the mitochondria says no? I'm not doing anything anymore.
So the mitochondria, and I already introduced that initially, are actually involved in far more processes than this ATP production. So, for instance, they also induce cell death, and they are involved in immune responses and so on. But let's say the prime readout that we would kind of look for initially in the mitochondria would be indeed whether this respiratory chain, so these protein complexes that are needed to make ATP, whether they are functional. So that's kind of the standard thing you would be investigating. And in Parkinson's disease, it's often this first respiratory chain complex that then is not functional, and you can measure this in certain ways. And another indirect readout is that if this respiratory chain is functional, then actually, so this organelle has actually an inner and an outer membrane, which is also kind of special. And over the inner membrane, this respiratory chain creates a proton gradient that you can measure. And there are dyes available that allow you to...
Green as well?
Can be green. That allow you to basically assess whether this proton gradient is functional or not. So that would be one kind of typical readout that we would explore in a neuronal culture or any glial culture to look into mitochondrial function. But then, since kind of maybe slightly more recent work is in Parkinson's disease is also going towards the role of mitochondria in inflammation. So another readout that we are now really kind of curious about is anything that is released from the mitochondria in a, let's say, pathological manner. So, for instance, you would not really necessarily assume that the mitochondrial DNA can be found anywhere outside of mitochondria in the cytosol of the cell. So only in some kind of disease state you would assume that this could happen. So we are looking for this mitochondrial DNA in the cytosol of the cells as a potential inflammatory trigger. So that's, for instance, also something we are quite excited about, because apparently when the mitochondria get dysfunctional, so typically they should be kind of cleared up by a process called mitophagy. So there the lysosomes come and they can kind of, depending on the membrane potential of these mitochondria, there is a cascade that then gets triggered, which basically labels these dysfunctional mitochondria, and then the lysosome recognizes a dysfunctional mitochondrium and basically degrades parts of it or even the whole thing. And if that process is disrupted, the result is loads of mitochondria that are not really functional, kind of spoiling the pool of happy and healthy mitochondria. And we are looking for phenotypes that go hand in hand with that, trying to understand, because mitochondrial dysfunction is one thing, okay, great, if you want to investigate that, and especially in genetic forms of Parkinson's disease, that if the gene and the protein that you are investigating are crucial for this mitochondrial function, then you should have a problem that kind of occurs more or less anywhere in any cell type. That still doesn't quite answer the question, why is it the dopaminergic neurons that are dying? So we're trying to understand if there's any of these processes that somehow make it more likely that a dopaminergic neuron has a problem. That's why also this interest in inflammation, because dopaminergic neurons cannot be regenerated. So they will mature during our embryonal development, and then you have a certain number, and then it only ever declines. And if somehow there was an inflammatory trigger, basically our immune system, our glial cells would even work against these cells, so against the dopaminergic neuron. And obviously you don't want to have that. So you want to have as many dopaminergic neurons as you can preserve. And so, if there is potentially a link to mitochondrial signaling, then, and we could influence that could be a solution to Parkinson's disease.
That sounds very promising and interesting as well. I understand why you get excited, because, it's one of those holy grails, let's say, and important also for Luxembourg, the research in Parkinson's disease. So in a second, let's talk Parkinson's disease, but you jumped a little bit with the mitochondria, neurons and brain and all that. So I was just thinking, again, very generally, when you look at mitochondria, do you really have to specialize in the cell that you look at with the special mitochondria? Because here we're talking brain and neurons, right?
So obviously, because our interest is indeed in Parkinson's disease, which is a neurodegenerative disease, we are primarily looking into cell types that you can find in the brain. And original work is around mitochondria was mostly done in dopaminergic neurons, if you can create those. And that, again, is a relatively recent development, thanks to Professor Yamanaka, who kind of came up with a protocol that allowed reprogramming somatic cells, such as skin cells, into any cell type of interest. And that can then be neurons, even dopaminergic neurons. So originally, this protocol was used to really specifically look into neuronal populations and what do mitochondria do there. And now, more recently, with this understanding that mitochondria might be so much more complex and that they could also potentially trigger inflammation. So then it starts also being relevant to look into, for instance, microglia, which are the immune cells of the brain, and to see, okay, so if I have a mitochondrial problem in the neurons with the same genetic defect, I may very well also have a mitochondrial problem in these glial cells, in these microglia. And what does this do? Does this mean that these are potentially more activated, that they lack energy? Moreover, even if you want to simply understand what the energy needs are of a dopaminergic neuron, then this is largely depending also on astrocytes, another glial cell type, because they're basically shuttling metabolites all the time to the neurons. So to really just look at one cell type in isolation may be practical, but it's really far from the reality. So in some kind of dream experiment, you would have to have all of these cell types in the same dish and then start studying what's happening to the dopaminergic neurons at a given time or with a given stressor. And so we are only really just getting there to make these co-culture systems where there is more than one cell type of interest present. And that's actually tough to achieve because each of these cell types has different cultural needs. And to somehow find a compromise there in terms of media that all of these cells can survive for a given time in the incubator is not simple. So while this is for us really just a medium, I mean, the cells are really, we are interested in the mitochondria, and yes, we have to do that in the right cell type. You are basically always touching on these other fields in biology because so that I can use neurons, that I can look into microglia, that I can look into astrocytes, I have to also stay ahead of what's happening in stem cell biology, what's happening with these protocols. Are they getting better? Are they allowing better combinations? Are they getting cheaper? So that is there a certain way that I can now create potentially purer cultures for less money, which is quite critical in science as well. And so you're all the time a little bit between the fields and trying to learn from all sides.
And on top of it, also, you have colleagues that are doing something parallelly, right, that you can learn from too.
Get inspired by, exactly. So, for instance, in my institute, there's one group by Professor Bambon that is mostly, I mean, yes, they also do Parkinson's research, but they are much more interested in the modeling aspect of it all. So they want to make their perfect iPSC-derived cellular model of Parkinson's disease. So they work on organoids, which are kind of 3D structures that are in a way trying to represent the brain. And so getting inspired by this work, we can potentially improve our, let's say, experimental landscape to answer the questions that we are interested in a more optimal manner, let's say.
And then it's also the type of PD you're looking at, right? Because it's not all, there was the discussion, I remember, when was it? 2 years ago, I think, when I had Dr. Ibrahim Boussard here talking about their breakthrough and the award and everything. And, I was all appreciative and saying, wow, this is amazing. Yes, but this is a very, very small genetic type of PD. And, the ocean is still there, right?
Ibo is my friend. So the genetic form that he talks about is a mutation in DJ1. It's this very, very rare familial form of Parkinson's disease. And exactly as he already points out, while we now have a kind of decent understanding, or at least slightly improving understanding of these genetic forms of Parkinson's disease, all of them together make only about 10% of of PD patients. So then in the remaining 90%, you cannot really say that a mutation in a certain specific gene causes the disease. However, also their mitochondria might play a role. So we actually, together with the professor of Ivo's team, and also Patrick May, so Reiko Krüger, Patrick May, and my team, we recently did a kind of, I would be so brave and call it cool study, where we looked into the abundance of common variants, so that kind of the difference between us is due to variants that are very common and a combination of different variants that are not disease-causing, because otherwise we would all be looking the same. And if you have a whole bunch of these common variants in a signaling pathway that is relevant for the mitochondria, let it be the creation of new mitochondria, the clearance of mitochondria, apoptosis, so whatever, something relating to mitochondrial function. And if you have a higher occurrence of such variants in one of these pathways, and we even kind of dissected them, and we found that an increase of common variants in genes and then proteins that are involved in how this respiratory chain works, that increases your risks to develop Parkinson's disease. So we looked into the PD cohort from locally from Luxembourg and controls. And basically in the patients, you find a higher abundance of such common variants, which are themselves not disease-causing, but the cumulative effect in such a pathway, and that was specifically in the respiratory chain, is associated with your risk of developing Parkinson's disease. And that actually then went further. So again, first of all, that goes to show that the mitochondria are relevant there. And then actually postdoc in the team from Reiko, and he was shared, and Giuseppe Arena, he then actually went to functionally study these cells. So we here, being super lucky with this cohort, we could actually go back to these patients. So first of all, we classified everyone and said, okay, you have a high risk, you have a low risk. It's called a polygenic risk. So if you have loads of these variants, you have a high polygenic risk in that pathway or a low polygenic risk. And so we actually found people with a high polygenic risk, such with low polygenic risk, and we grouped everyone, and then we went back and we got their skin cells and we made stem cells and then neurons. And then Giuseppe actually studied the mitochondrial function in these different cultures, and he did indeed find correlates. So that meaning that if you had a high polygenic risk in the respiratory chain proteins, then this also really did have a functional consequence. So with this understanding, you could potentially explain also what could be a contribution to someone having a sporadic form of Parkinson's disease. And maybe there the mitochondria are not the non-plus ultra explanation, but they are a contributor and potentially something else that happens in the person's life. I don't know, whatever other hit, an environmental hit, some kind of exposure to something. And then this
unknown
together...
chemicals, for example, that Professor Szymański studies.
Exactly.
Right?
Actually, we also have a study with Emma Szymański on trying to understand whether there is an interaction between such polygenic risk and environmental factors, and then whatever other hit occurs, and then this person develops Parkinson's disease. But this is really just the beginning, but it's a cool beginning because you could potentially use this genetic information to stratify people and could already in a way predict, and that was another arm of the study. We retrospectively looked into a clinical trial where a mitochondrial drug was tested. And we checked if people who had a high polygenic risk score in this pathway would actually be the better responders to that. And that you would expect that, because if you assume that indeed there is a link between this genetic profile and your mitochondrial dysfunction, and then you get a drug, then you should be more responsive to that drug. And that indeed was the case. And so that was all in very small numbers of patients. So now we would like to do that in a larger way, and we are currently kind of looking for funding there internationally, and maybe even from the Michael J. Fox Foundation, to kind of understand this better and possibly use this as a stratification tool for clinical trials and for to better group this huge number of PD patients where we don't know what's going on.
Okay, so fingers crossed for that, for sure. But just, if we say that mitochondria play a big role and their dysfunction plays a big role, What about the possibility to cure them? Can we cure them? Can we transplant them? What do we know on that side of things?
But that actually relates to what I just said. So because if indeed, actually the assumption is not that everyone has a problem with mitochondria. The assumption is that a certain subset of patients has primarily a mitochondrial problem. So now if you go and just treat any patient with a mitochondrial drug, you may not overall really get a good response. But if you were to know who is the subset of people who really have a mitochondrial problem, then, and you go in with treatment, and not at a time when they already have Parkinson's disease, but ideally you go in and treat before they even lose their dopaminergic neurons, that is kind of the dream solution, because you cannot now come and when 50% of the dopaminergic neurons are gone and boost with, I don't know what, energy providing drug, that's too late. So in some way, the future cure needs to look like that you would have to have some kind of genetic prediction of who gets Parkinson's disease, and then you go in and you treat that person ideally with something that's not harmful, so that you don't have an ethics problem that you treat a healthy person with a drug at a time to prevent this degeneration of dopaminergic neurons in the first place. But that's obviously a very complex topic. But that's where the mitochondrial dysfunction basically could be the solution for treatment. Yeah, right now with the current kind of treatment regimes, it's going to be almost impossible, I would say. So really, we need to learn more so that we can, without risks, treat people at an earlier stage.
So we have loads of different points in time that are happening closer, further, funding dependent as well, for sure. And the last point that reminds us, of course, to go back to our pub quiz question that you mentioned a long time ago. So if you can remind us what the question was about and then tell us the answer.
So I explained to you that the mitochondria, these powerhouses, that they contain their own DNA, and which is a circular structure, a little bit reminiscent of bacterial DNA. And my question was related to how many copies of this DNA do you assume is present in a regular cell?
And the answer is?
And the answer is that it largely depends on the cell type, but that it's in the area of 1,000 to 2,000 copies. So significantly a higher abundance of these genes per cell to any other nuclear gene that you would find in the cell, which is interesting.
But what does it mean then? That many copies, what do they influence? What do they change?
I mean, the metabolism will be adjusted to the amount of this protein that then can be produced from these copies. It's from a genetic aspect, it's quite relevant, because it means that a mutation that occurs in this DNA is actually largely irrelevant until it is present in a vast amount of these copies. Only then it has a functional consequence. And that's something in the area of, I think, 70-80% of the mitochondrial DNA needs to contain the same mutation, so that then the impact of this mutation on the protein landscape that is being encoded by this DNA is so affected that you actually get a physiological problem. So this threshold is quite variable because of the vast amount of mitochondrial DNA, and it also has something to do with how these mutations are being transmitted. And there are different theories that there can be a situation where basically a mutation somehow suddenly takes over the entire mitochondrial DNA pool. So people are trying to basically study this not so much in the context of Parkinson's disease, but in mitochondrial diseases, which are really, really terrible diseases. What can influence a shift from, let's say, a mutation amount of maybe 20% to suddenly 70-80% in a cell, and that's in these diseases really relevant in terms of fertilization. So if in an oocyte you suddenly have 70-80% of a mutation present that in other circumstances was only present in maybe 10-20%, then that obviously creates risks for potential offspring. And so these mechanisms that are behind this mutational shift, they're something that's kind of interesting to understand. And there is a link to Parkinson's disease because work from our lab actually showed that in the mitochondrial DNA, you also see an accumulation of mutations in Parkinson's disease. Like it's just per se, throughout the entire genome, you find accumulation of certain types of mutations, but not the one mutation which suddenly increases massively in abundance, but it's kind of the overall, we call it somatic mutational load is simply increased, which might again reflect that the mitochondrial quality is simply not great in these cells in Parkinson's patients.
So yet another path to look into for sure. I hope that our listeners today, if they are, choosing the study subject, they got inspired and they will just go into neuroscience or, any kind of study of what you're doing at the LCSB. Listeners, if you want to know more about what we talked about, so the interview I had with Dr. Ibrahim Boussad, check it out. It's very, very interesting. Also, as I mentioned before, Professor Emma Szymański was there too. So the other side of LCSB's work and the unknown chemicals. And we had Dagny Aurish as well. Quite a few different people from LCSB to check out because, of course, we can't discuss everything with Anne today. But what remains is to thank her very much for today. Thank you, Anne. This was really, really interesting.
Thank you again for inviting me and for giving me a possibility to explain a little bit what we are doing here, actually, which is quite connected also to the work of each of these colleagues that you just mentioned.
Exactly. So the pleasure is all ours. This is it. And if you want to know more, you can always check what we are writing on all the social media. We are there. And our podcast is present on all the major podcasting platforms. This was SciLux, and my name is Hanna Siemaszko.